you for joining the inaugural seminar on mathematical and computational modeling of cancer, or MCMC for short. I'm very happy that our first speaker is Rob Noble. He obtained his master's in mathematics at the University of York, his PhD in mathematical biology at the University of Oxford. He was a postdoc in Montpellier and ETH Zurich, and now he's a senior lecturer in applied mathematics at City University of London. And he'll be talking to us about understanding, predicting, and controlling stochastic cancer evolution. Thank you, Rob. And before you start, please post questions you have in the YouTube chat. You can post them during the seminar or at the end. And do let us know if there's something that is not working. Thank you.
Thanks very much for the invitation, Ivana. It's an honor to present the first of these, what I hope will be a long and successful series of seminars. So I'm going to talk about some very recent work, which we have up as mostly as preprints, some of it not even up yet, on this topic of understanding, predicting, and controlling stochastic cancer evolution, which is the main theme of my research. And a key word there really is stochastic. And that I'll show you the underlying, why that is an important underlying theme. So another subtitle for this might be Poisson versus cancer. Ivana and I were just talking about how we both teach stochastic processes, and having taught a lot about Poisson processes for a few years, I was keen to bring them a bit more into my research, and you can see that as a theme. All three of the projects I'll present overviews of here are really built fundamentally around Poisson processes and what happens if you extend Poisson processes in various ways as relates to cancer. So the starting point here is that we see cancer through an evolutionary lens. We see cancer as an evolutionary process. This is a classic figure that many people in my field start their talks with. I'm not going to go into the details of exactly what Peter Nell meant here, but essentially what he was proposing nearly 50 years ago was that we can think of the development of tumours into cancer as an evolutionary process that can be represented by a tree, starting from a single normal cell and then developing into first a small tumour and then later on an aggressive invasive cancer. And so this is really the starting point for everything we're doing. So the amazing thing is that these days, instead of just drawing these cartoons, as were done 50 years ago, we can look at actual trees of tumours that have been removed from patients and then submitted to some form of sequencing. In this case, these are kidney cancers that have been analyzed by the TRACER-X consortium, a huge project in the UK. And what they did is they removed these tumors and then they did multi-region sampling. So they removed several samples from different parts of the tumor. They sequenced each one, and from looking at which mutations were present in each sample, they were able to construct trees, and they were able to construct a lot of trees. So these are examples of some of those trees. Now, this is from 2018. There are more recent data sets, which we also now have access to with hundreds of patients. This is a fantastic resource. And what you can see here is that in the layout of these trees from top to bottom, there is something of a pattern. They're split here into rows, which are stage 1, stage 2, stage 3, and stage 4, as I've highlighted here. And the general pattern you might notice is that as we go down from stage 1 to stage 4, the trees tend to get a bit more complicated, which means the tumors are getting a bit more diverse. The trees are getting more branched. And that is something that the TRACER-X team expected to see, and in fact was one of their pre-registered hypotheses that they wanted to test. And that's something that we also wanted to look at to see if we could understand the nature of this relationship. So here's the pattern that the TRACER-X consortium reported. What they did was they applied an intratumor heterogeneity index, which basically looked at how branched the trees were in some sense. And they split all their cancers into two groups, one with low diversity and one with high diversity according to their index. And what they found, this is a survival plot. So the steeper these curves go down, the worse it is, because every drop of this curve corresponds to a patient progressing either to more advanced cancer or to dying. So this is called progression-free survival. At the beginning, the whole cohort is treated, and then we follow them over time. The TracerX consortium followed them to see how they survived. And what you can see is that the high diversity cohort, those with more heterogeneous tumors, survived less well. They had shorter predicted survival times. Now, if we look at the statistical significance, though, these p-values, what it tells us is that, yes, this is obviously a significant pattern, but if they adjusted it for the standard clinical staging and grading methods that are used to put patients into cohorts and predict their outcomes, then once they adjust for those standard clinical markers, they find that this pattern is no longer significant. So although the pattern is there, it's not really adding anything to the ability of the clinician to stratify these patients. Now, we wanted to dig into this and see maybe we can find some better indices. We can extract more information from the trees than they could with this basic index, and we could understand more about how we can stratify these patients. So why did we choose to do this using stochastic processes? Well, in a lot of mathematical oncology research, particularly in terms of looking at cancer evolution, there are a lot of models that are based on ordinary differential equations, which I've abbreviated here to ODE, or partial differential equations, PDE. And these models are nice because they're very easy to construct. They're very simple to run numerical analyses of using standard software. They can be tractable, which means that you can find for simplified models, you can find approximate analytical solutions, which gives you insights into why the model behaves or how it does. They tend to be parsimonious. You can construct these models without very many parameters. So these are very popular models, but a drawback is they're not stochastic. So they cannot capture some of the properties that are inherent to evolution, particularly drift, which is random changes in the frequencies of different mutations in the population, or stochastic extinction, which means that when a population size becomes small, it can become extinct just through bad luck. And these kind of behaviors are not captured with ODE or PDE models. On the other hand, we can run agent-based models. So these, again, are very popular. They're quite fairly straightforward to construct. What we do then is we write a lot of computer code, and we have every cell or groups of cell in a model, and we have them follow some rules which have some random components to them, and we allow a tumor to grow according to a certain set of rules, and then we run a lot of simulations to find our results. Now, this is nice because this approach is stochastic. You include a random number generator, you have the cells behave stochastically. But all of these models, especially when you include spatial structure, they tend to be less tractable because we don't usually have very good ways of analyzing complicated behavior in these models, and they're less parsimonious. You have to put a lot of different rules and assumptions into these models to get out the kind of behavior you want, and someone else might approach the same problem with a different model. So the nice thing about stochastic processes is they tick all these boxes. They can be tractable, parsimonious, and stochastic. And so that's really, I think, they give you the best of all worlds. And what you'll see, though, is that we are not just relying here on stochastic processes. The nice thing is you can construct a stochastic process, and then you can look at what is the deterministic ODE counterpart, and what is the agent-based model we can use to test the validity of our predictions. So you can actually take all three approaches and you get the best of all worlds. So this is the basic setup of all the recent projects I'm going to present, is that the tumor cell subpopulations generally are growing deterministically. So we have deterministic population growth, but mutations new mutations are arriving according to a Poisson process. And with that basic setup, you can extend this in different ways. You can look at different complications here to analyze, well, to answer different questions or test different hypotheses. So the first version of this kind of model we're going to look at is one that adds spatial structure. And this is inspired by a figure that I used to start many of my own talks, which is a cartoon essentially from a review from a few years ago that was proposing different ways in which tumors could potentially evolve. So in these diagrams, from left to right, we have a progression of time, and from top to bottom, we have the proportions or the frequencies of different genotypes within the tumor. And these are different genotypes of interest. So not necessarily all the genotypes, but just whichever ones you most care about, the more interesting ones. And in this review, they proposed 4 different ways in which tumors might evolve. So in the first case, we get punctuated evolution, which means that all the interesting stuff happens early on. We get a few selective sweeps, which means that some mutations manage to spread through the entire tumor because they are beneficial to the cells. They give them higher fitness, more competitiveness. But then after that, there's pretty much stasis, and the tumor doesn't do very much in evolutionary terms. It just grows outwards. The second case is branching evolution. Well, all of these evolutionary modes eventually are branched if you look in enough detail, but what they're talking about here is some sparse branching. So if you look at the tree, it will have a few big branches, and these branches will only occur quite late on during the tumor evolution. So you get this kind of classic evolutionary tree. The third case is linear evolution. Here, again, we have these selective sweeps, which means that each color here is a new genotype that is replacing what came before because it has some advantage over it. So if you were to look at the tree in this case, it would be a straight line. You would just get a linear tree, no branching. And the final case is neutral evolution. In this case, we see a lot of branching and we don't see a lot of selection going on. We don't see types replacing each other very extensively. We don't see these sweeps occurring. So you just get more and more diversification. And this could be truly neutral or just effectively neutral evolution, which means effectively neutral means that there might be some local competition and selection, but globally, there's very little. So the question then is which of these is really prevalent and which of these should we expect? And this is an important question for prognosis, for predicting what's going to happen with the tumor and for developing an effective treatment strategy. And this is, again, some data. So this is from a 2019 paper, which looked at 3 cancer types. And they were looking particularly here at BRAF mutations and the shape of evolutionary trees in these tumors from a large database. Now, there are some caveats about this approach, but what they found is that there are different degrees of linearity or branching within these tumor types. So particularly SKCM, which is melanoma here, tends to have more linear trees than the colorectal and lung cancers. And so it's interesting to think about why that might be. And also it sets up this question for why is it that there is a certain proportion of tumors that have linear trees and the rest have more branching trees? Why do we get this variety of tree shapes? Can we get some kind of grasp on why this should happen? So the way I approached this was with a very simple physics type model where we simplify things a great deal. We model tumors as balls in 1, 2, or 3-dimensional space. And we assume that space is continuous. So we model the growth of a tumor from a point to a ball as a growing ball growing continuously with time. So the initial type here, what we call the wild type, which is the initial type of cells that are present in the tumor, these are shown in a kind of pale yellow here. And the ball of the wild type tumor grows at a certain speed, which we call CWT. And this is a constant radial expansion speed, which means that the radius is growing at a constant speed over time. This gives polynomial growth. In 1, 2, or 3 dimensions, you get linear, quadratic, or cubic growth of the entire volume of the ball. Now, this is of course, a simplified model, and we'll look at different versions, different alternative growth models later on, but this one is a particularly nice one to analyze. And what we then assume is that as the world type is growing at any time, we can have a mutation occurring, and that is shown here in blue as a little blue star. So the mutant arises at a random time and at a random location within the yellow ball, and the mutant grows at some other speed, cm. And we generally assume that cm is larger than cwt because that makes this an interesting problem, which means that there is some possibility that the mutant, the blue one, grows to complete a selective sweep and completely replace the original type. So the blue ball must grow faster within the yellow ball than the yellow ball is growing into whatever surrounds it. So we have cm greater than cwt in the interesting case. And the question then is, how likely is it we get a selective sweep, so the blue type completely replacing the yellow, versus the alternative, which is that another mutation, a green one here, occurs within the wild type and survives before the blue one can attain a sweep. And then what we get is what's called clonal interference. The two mutants compete with each other, and you're more likely to get a branch tree in that case, as shown on the right here, than a linear tree, which is what you expect in the case of, what you inevitably get in the case of the selective sweep. So we're looking here at the first part of the evolutionary tree, not what happens further down, but what is the first event? Is it linear or is it branching? And the nice thing about this model is it has very few parameters. So the parameters are the speeds, cm and cwt, the mutation rate, mu, so how often these mutations occur, and the spatial dimension, d, if you count that as parameter, which will be 1, 2, and 3. And what's very nice about this is that you can analyze this model. You can analyze the exact model, which Alex Stein here, a very talented graduate student, did. But also what Alex found is you can find some very nice simple approximations by making what seems like a very radical simplification, which is to assume that instead of the first mutation arising anywhere at random within this growing ball, it occurs exactly at the middle, exactly at the center of the ball. And that seems like a very crude assumption, but it turns out that if you make that assumption, you get much simpler mathematical results, and they turn out to be, as shown by the dotted line, sorry, the dashed line versus the solid line here, very close to the results of analyzing the exact model, the complete model. And so you get these nice results that for these different curves here showing different properties, the most important one being in the middle, which gives you the probability of a sweep versus the ratio between these two speed parameters, cm and cwt. So this is how much faster the mutant is growing versus the wild type. And what you find is that you can approximate the probability of a sweep with this simple expression, 1 minus cwt divided by cm, all to the power of the spatial dimension, which is a result we thought was rather elegant. And then what we did is what Marcek back here did was to run a huge number of simulations of this system using a spatial Moran process, and many thousands of simulations on a cluster computer. And what he verified is that our predictions are very close to the results of these simulations. Now, something else you might notice here is that this result for this probability of a sweep, it doesn't include the mutation rate. So this is a very interesting result, we think, that the probability of a sweep is entirely independent of the mutation rate. And this is verified here by the simulations, which you see here over 2 orders of magnitude, different mutation rates, you see consistent results. And this is, we were able to argue why this is the case, but this was somewhat unexpected when we started. This applies in 1, 2, or 3 dimensions. So in summary, what we find is that if you parameterize for cancer, if you put in parameters that are kind of ballpark for tumors, which are on this slide here. So I'm not going to go into these, but I hope most people agree these are reasonable ranges for tumor parameters. What you find is that if you observe a sweep in a tumor, or in other words, if you find that the first part of the tree is linear, then we can deduce that that sweep probably began before the tumor reached around 400,000 cells, which is a very small tumor, and it probably ended before the tumor doubled that size. And these results are very robust to change in parameter sizes. So what we find is that selective sweeps are rare, except during very early solid tumor growth. This is an analytical model that gives you that conclusion. And we did look in, if you look at the preprint, you'll see that we looked a lot at alternative growth models as well, and they generally predict lower sweep probabilities. So what we were looking for here was an upper bound, and that's what we obtained with this kind of model. So I've shown you a bit of trees, and I've talked a bit about in those slides about tree shape. And so when you start running these models and your main output are trees, then you start thinking about how do we summarize these trees? How do we summarize the shape of these trees? And this led me to an entirely different area that I never expected to get into, but ended up being necessary for interpreting the results and being able to think quantitatively about these tumor evolutionary trees. So what we want to do then is quantify tree shape. And this will allow us to categorize and compare trees in some meaningful way. And for these to be good indices, what do we need them to have? Well, there are different kinds of indices that have been developed in the literature. They generally fall into three categories. The first one is measures of diversity. So effectively, how many leaves or tips does your tree have? The second one is some sense of bushiness. So what is the average out degree in some sense of the tree? Is the tree simply bifurcating? Every node has exactly two daughters, apart from the tips, or is there a greater degree of branching than that? And the third one is tree balance, which basically means that it is when there is a branching event, how evenly sized are the subtrees that come from that branching. So as I say, there are many indices of these types that have been proposed, and the hazard is always what is shown here, which is that you say, there are so many types, let's propose a universal system that replaces all of them, and you just end up with another alternative. So this is not what we wanted to do. The reason I started working in this area is because we found the existing methods were not fit for the purpose we wanted. So really what you'd want to have useful indices for particularly this problem of looking at tumor evolutionary trees, we need indices that are robust, which means that they are insensitive to small changes to the tree. This particularly means that if you have different sequencing sensitivity, you have different ability to detect very rare mutations, then these indices won't change very much. The second one, universal. We want to be able to apply these indices to absolutely any rooted tree, not conditional on what is the degree distribution or various other properties of the tree. And the third one is we want these indices to be interpretable. So we can meaningfully compare any pair of rooted trees. And where we see an index value is larger than for one tree than the other, we can explain in words what that means in general terms. And what we discovered is that none of the existing indices fulfill these three properties. So that we started from these three properties and said, how can we develop indices that have that fulfill all three of these? And so what we ended up with, thanks to again, very talented master students they were at the time, Jeanne and Cécile, is an index that is a weighted average. So the index here is called J1, and it is defined in terms of, for each node in your tree, which is labeled here I, you assign an importance to that node, which is dependent, in fact, it is simply the sum of the size of the subtree that descends from that node. So basically, how many descendants does a node have? That determines how much importance you give to it, which means nodes higher up in the tree are assigned more importance. That ensures robustness. The second part is a node balance score, which is a way of assessing how evenly that node splits its descendants into equally sized subtrees. And what we do is then simply take a weighted average. So the bit at the beginning here is just a normalizing factor. So we end up getting something between 0 and 1. It's just a weighted average of these node balance scores across the tree. This seems like a very obvious idea, but it actually took us a long time to discover that more than 50 years before us, well, almost exactly 50 years before us, a computer scientist called Niebegelt and his colleagues discovered essentially exactly the same index and proposed it in the context of looking at binary trees for information retrieval and storage in computer science. But that discovery was completely forgotten about until we eventually managed to dig it up many years later. So what we're doing is we're saying this idea, originally Niebegelt's, that we've rediscovered, is a very useful way of looking at tree balance. In fact, we argue it's the best way of looking at it. So as I say, SI here is the sum of the size of all descendants of node I, and WI is the normalized Shannon entropy. This is the way we choose to measure the balance of the node. So this all descends from the Shannon entropy, for those of you who know a bit about information theory, which gives it a lot of nice properties. So here's an example calculation. What you do is for every node in your tree, you assign an importance score, which is the S value here. And you can see is greater for the tree, the nodes higher up. Each node here, the numbers inside the nodes are sizes that are assigned to those nodes, which in cancer terms, we generally assign these sizes as the size of the population corresponding to that node. So each node in a tree like this corresponds to a clone or a genotype within the tumor. And if we know its population size, we can assign a size to the node, and those are what are inside the circles. And then the W values here are the balance scores. So what you do then is you simply take a weighted average. And in this case, this tree gets 0.89. So what we then did, we
Sorry,
found
Rob, before you continue, maybe you said it, but I missed it. A larger score, what does it mean?
Oh, yes, sorry. So the larger the value of J1, the more balanced the tree. So a perfectly balanced tree gets a score of 1, and a perfectly imbalanced tree scores 0. And then any tree you can come up with will be assigned a score somewhere between those extreme values.
So for this tree, this is a high score. So this is pretty balanced.
This is a fairly balanced tree. If you look at the root node here, it gets a score W equals 1, because if you add up the sizes on the left-hand branch, 4 plus 1 plus 3 plus 3 plus 3, you get the same as if you add up the right-hand branch, 8 plus 5 plus 1. So you get 14 in either case. So the root is perfectly balanced. And here, this one's almost perfectly balanced because it's got 8 on one side, 6 on the other, which is close to equal. This one is less balanced because it's got 1 on one side and 9 on the other. This one is perfectly balanced because it's got 3 and 3 and 3. So overall, when you take the weighted average, you get something that's pretty high.
I see. And so one more question, because I'm really interested in this.
Oh.
So based on what you're saying, it seems that you calculate the sizes of the population on different sides of a node. Do you take branch length into account as well?
I'm just coming to that. You've
Okay.
exactly you preempted my next slide. So this first index we came up with, it takes into account the topology of the tree and the sizes of the nodes, but it ignores branch length. And so then a problem that bugged me for ages is, how do you account for branch lengths in a nice way? And it took me a long time to come up with it, but finally I came up with a nice way of doing that has all the properties you'd like it to have, which means that it varies continuously as you vary the branch length. And as you shrink a branch length to 0, then the index behaves the way you would. It continuously moves to the value you would expect it to. And so what I did then was I found in doing that in fact this balance index can be related to a diversity index, and all of these three different aspects I talked about, the diversity, the bushiness or the effective average out degree, and tree balance, all of these can be seen as variants of the same idea and can be cast within a single system. And then we can look at, so this way of averaging by the node importance score is one way of averaging across a tree, but there are other ways. Another way to average it is going down the tree and looking across the tree. You can do what we call the longitudinal average rather than node-wise average. So you can do that for each of these three types of index: the diversity, the bushiness, and the balance. And what you form then is this universal system, which is mathematically really neat, because all of these indices, you can put them in a grid, as I will just show you, and they capture all these different aspects of tree shape in a very pleasing way. So the basic idea is that in some way, we define indices as weighted means of basic diversity, and evenness indices, accounting for both node sizes and branch lengths. And what we show is that this method is very robust. It's applicable to all rooted trees, and this system unites measures of diversity, balance, symmetry, effective height, effective out degree, effective branch count in a coherent system. So all of these different ways of looking at a tree, they can all be put within this single system, which is summarized in this table here. So what we see as we go across here, we look at these three index types: richness, which means you ignore the size, you just look at the count rather than the size. A diversity index, which tells you in some sense how many of something there are in an effective way. So the first one tells you how many there are just counting. Second one, how many there are accounting for their sizes. And the third one, evenness. So how even are these things in terms of their sizes? And what you can do is you can just discard all the tree information, and then what you recover are classic richness, diversity, and evenness indices, such as Shannon diversity. If you average down the tree, across the tree, rather, as you go down it, then what you get is the longitudinal mean, and this gives you ways of looking at the average branch count across the tree, or the effective number of maximally distant leaves, or the evenness of branch sizes across the tree, which can relate to symmetry as well. If you take the node-wise mean, which we were talking about previously, then you get measures of average effective out degree, so the bushiness of the tree, or tree balance. So the diversity becomes the bushiness measure, and the evenness gives you tree balance. And then finally, what you can do is you can take all the branches, reattach them to the root, so that you look at them all independent of where they are in the tree. And in that case, you can get the effective number of non-root nodes, you can get the effective number of branches, and the evenness of all the branch sizes, ignoring where they are in the tree. So this is, I think, mathematically elegant, and we go into a lot more details in this preprint about that, how that is so. But the big question then is it actually useful? You can develop a nice mathematical theory, but what's its practical use? So the nice thing is that I developed this new system before we applied it to any data. And so having developed these indices, we then said, what data sets can we apply it to? And the data that came along, and you can see here, it was actually published after we developed our methods, was in a new paper from TracerX by Frankel et al. And the great thing about that paper was they had far more trees than before. They had the clinical outcomes, and they made all the data public, which was immensely valuable. So I'm very thankful to them for doing so. And what Kim here, my PhD student, did is she took all of those trees from actual tumors, and she applied our balance index, the one that accounts for the branch lengths as well as node sizes, and she assigned scores to all of these trees. So here you see examples of highly balanced trees and relatively unbalanced ones. And then she saw whether that was predictive of clinical outcome. And what we were amazed to see is it really is. So this was quite startling. So here we see three categories: highly balanced trees, intermediate, and unbalanced, according to the index that we developed independently of this data set. I just want to emphasize that again. We had no ability to fiddle this because we used all the data and an index we developed independently of this data set. And what you see is there really is some predictive power here. And this is true also if you regard this balance index as a continuous variable as well. So it's not just that we cherry picked the three categories here either. And what's more, Kim showed that even if you control for staging and grading, the standard ways of stratifying patients, this still has predictive power. So it adds to what clinicians can currently do.
And this is super interesting. And I would like to know, how do you understand this connection between survival and a tree balance?
We don't fully know. So I think this is what is, it is one of the things that we are currently investigating, that Kim and others I'm working with are doing at the moment, which is trying to develop models that can explain this pattern. So we have hypotheses, but we really need to test them with mathematical and computational models, and then compare that further with the data to really get a handle on why this is happening. We have some hypotheses, but at the moment we don't have anything that we have really confirmed. But I'm sure you have your own hypotheses based on your own fantastic work in this area. So
I mean,
yeah.
My hypothesis would be that the unbalanced trees have some kind of subclonal selection going on, and maybe they're more aggressive due to that.
That's our main hypothesis. That this is a proxy for the extent of selection going on in these trees. And so that's why we've been running a lot of models with and without selection to see how that affects the balance of the tree and our ability to detect selection using our indices. So that is something that Kim and others have been pursuing to try to understand these patterns. Something else we're looking at, of course, is different cohorts, different cancer types. And the pattern seems to be more complicated. So that this works well in the TracerX cohort, but in other cohorts, we're trying to understand different patterns. And that's not unexpected because we expect different evolutionary dynamics in different cancer types.
And one more question I have about this is, so if I understood your table correctly, you can basically pick an index based on which feature you're interested in. And here you are picking the balance.
Yes.
So what if you, why did you pick balance? And what if you picked another feature?
So Kim has gone into this very extensively. So she's looked at all of the indices, and she finds that, yes, the diversity indices are also predictive, but tree balance seems to be the most predictive. And there is some correlation between balance and diversity as well. So this is the one we particularly focused on, but the diversity indices are also useful. And that's not unexpected too. But we think balance is particularly useful because we think it could be detecting selection, and it's less obvious how diversity indices would be linked to a signature of selection.
Fascinating, yeah.
So the nice thing about these indices as well, I said that they have nice mathematical properties. Well, one thing you want to know is, how does this compare to a null model? And the classic null models that are used in tree generation are the Yule process and the uniform model. So the Yule process assumes that each leaf or tip has an equal probability of dividing into two at any point in time, whereas the uniform model assumes that all trees on a certain number of leaves are equally probable. And so for other tree shape indices that have been developed over the years, people have found the expected values under these null models. And so my former PhD student, Vess, who is now pursuing a postdoc, and an extremely talented undergraduate, Aman, who is at Johns Hopkins, and will be an asset to whoever he works with next. They we're able to analyze the properties of our index and obtain approximations to the expectation, which are very close. So basically, we can, if you give us a tree, we can say whether it's more or less balanced than a Yule or a uniform. You would expect, according to the Yule or the uniform null model, for that given number of leads. So this gives us a way of, it gives us kind of a benchmark for these kind of analyses. So in conclusion, these new indices, they allow you to compare any trees. You can do parameter inference, model selection, hypothesis testing with them potentially, and we can apply them to all kinds of other systems. So we're also applying these to trees of life, to all other trees we can get hold of to see what we can get out of them. So finally, relatively briefly, I'm going to talk about another project, which again is based on stochastic processes. So the link in the previous one is that essentially the Yule process is this kind of stochastic process. The arrival of new branching points is Poisson distributed. So our null model is essentially derived from a Poisson process. What I'm going to talk about now is a model of treatment. So the question here is, how should we treat tumors given existing drugs or existing therapies in general? And the general approach that's followed in the clinic is that the tumor is treated. If the treatment is effective initially, then the tumor decreases in size, as shown by this black line here, going down with these black points. And then the doctor essentially waits to see what happens. So if they're lucky, then the tumor seems to disappear and the patient is monitored in the long term. But what often happens is that the tumor eventually, although it became undetectable for a while, it grows back. And when the doctor sees this, when the tumor is once again detectable, they apply a second treatment and try to debulk it again, and this process continues. Now, it has been suggested, particularly by Bob Gatenby at the Moffitt Cancer Center, that a better strategy might be to treat before the tumor becomes detectable again. So if we know that this particular cancer type is often incurable with the initial treatment, it often grows back, then instead of waiting for it to grow back, wouldn't it be better to treat perhaps earlier before you can even see that it's growing back, just to be on the safe side? And would that give us a higher cure rate? Now, Gatenby and colleagues a few years ago, what they showed in a proof of principle model was with simulations, they showed that this is a plausible approach, right? They did essentially a proof of concept, but they had only numerical results. And what we wanted to see was, can we get some analytical grasp, a better theoretical grasp on whether this approach is likely to work? So the basic idea is that we have what we're calling N-min, which is the minimal tumor size, the nadir here, which is shown in orange. And we can treat either before or after that point, and we can treat at different times before or after. So these blobs here show us different times when we might apply a second treatment after the first one. And the question is, which of these is optimal in terms of maximizing the probability of curing the tumor with these two treatments, two strikes? So this is the setup. Again, Arman has been involved here, but the project was initially led and has really been the visionary behind all this is Srishti, who was she did a master's project with me. She is now pursuing a PhD in Heidelberg. And so the model that Srishti constructed is that we have a tumor which has a population size shown here. And we assume initially this tumor comprises some susceptible cells, marked here as S, and some cells that are resistant to the first treatment we're going to apply, R1, and some cells that are resistant to the second treatment we're going to apply, R2. So this is a heterogeneous tumor with three types of cells within it. And at time 0, we apply a treatment that is treatment 1. So this kills cells apart from the R1 cells. And during this time, because the susceptible cells are the majority, the tumor decreases in size down to N-min. But also during this time, we see further evolution, which means that we can have mutations from the R1 cells to R12, which are resistant to both treatment types, and from R2 also to doubly resistant cells. And then what we do is at some point, we introduce a second treatment, and we can apply it at any of these blobs. So either before N-min, at N-min, or after N-min, switching after N-min. And the question then is, what's the probability that we get extinction, this downward dashed line, versus this rebound here? So this shows you what would happen in the absence of a second strike. And what we want to do is make sure that instead of getting this rescue population here, we get this downward trajectory. So the main thing we're interested in is, when do we apply the second treatment? And the way Srishti modeled this, which was quite ingenious, is to use a deterministic model during the application of the first treatment, and then to use a stochastic model in the latter stages. And what she and Aman were able to show is that this actually gives you a very good approximation to a fully stochastic model. So the deterministic model is described by some differential equations, which are here. I'm not going to go into detail, but this is a fairly standard model here that has some growth terms with some density-dependent growth, and then it has some mutation terms. And this diagram here summarizes all these flows between the different compartments: susceptible, resistant to first treatment, resistant to the second, doubly resistant. And the stochastic model involves so we have a treatment switch time here, which we call tau, and we have a probability of extinction, PE, which is the product of two components. The first one is the probability of tumor extinction due to the standing genetic variation. That is the resistant mutations that are present when you start treatment. And the second one is the probability of extinction due to de novo mutations, those that occur during the treatment. And what is very nice about this is that Srishti was able to find from the evolutionary rescue theory literature ways of calculating these two probabilities of extinction. And this is essentially based on a Poisson process. And the idea of evolutionary rescue theory usually is that we're trying to maximize the probability of a population surviving some kind of adverse effect, such as climate change. And the question is, if a population is decreasing, for example, due to climate change, can it evolve fast enough to become resilient to this new environment and bounce back? And that's usually what evolutionary rescue theory is about. Here we're using the same theory to ask the opposite question. What we want is the extinction scenario. And so these are the form of the solutions that Srishti was able to find. So the nice thing about this project is that we had this very robust methodology. We had analytical solutions of this approximate model. We had extinction probabilities using evolutionary rescue theory. We had agent-based modeling as well, based on computational simulations. And Srishti did very extensive sensitivity analysis. So we really covered all the bases there. And the main finding was that the optimal time to treat is indeed close to the nadir. So close to this value N-min, where the tumor as it is at its smallest size following the first treatment. That maximizes the extinction probability very generally. The other question you can ask is, given that generally in the clinic, you cannot tell exactly when the tumor is at its smallest size because it's undetectable, is it better to treat slightly earlier or slightly later? If you can err on the side of caution one way or another, then what do you do? And what we found very generally, not in all the edge cases, but in all the main cases we looked at, is that it's better to treat slightly after the nadir, as you see here in blue, than before. And the reason for that is shown in the second set of slides here. It's down to this R2 population. So if you treat after, then you give more time for this R2 population that is there initially to be depleted before you apply the second treatment. So if you assume that R2 cells are absent at the beginning, then that difference between the two cases is much diminished, as shown here. So as I say, Srishti and Aman did many other analyses, supported to a very large degree by my collaborator, Yannick Viosa. And what they found is these additional results. I'm not showing figures just in the interest of time, but the main other results are that, interestingly, higher treatment efficacy is not necessarily beneficial. So if you have a less effective treatment, particularly if you have one more effective and one less effective, then you can still apply this approach and you don't do that much worse than when you have two very effective treatments. A cost of resistance is not necessarily beneficial. You don't necessarily need to assume that these cells are paying a cost for being resistant to one drug or the other. Carrying capacity, we include it in the model, but it doesn't actually have very much effect, so you can neglect it, and that simplifies analyses. The extinction probability increases with the death rate and the turnover parameters. And very importantly, this two-strike therapy, although we show that it can be effective, what we are assuming here is that the initial tumor size is quite small. So this might be the case if you are treating particularly metastatic cancer and you're treating small metastases, or you're treating residual tumor after there's been that the main tumor has been resected or whatever. But we really need to extend this work to look at multiple strikes. Our hypothesis is that what you could do is just repeat this. So if two strikes are not enough, you've reduced the size of the tumor enough that then you can apply a third strike, and that might be enough, or a fourth strike might be enough. So what we expect is that if you have larger tumors, then multiple strikes would allow you to drive the tumor to extinction, although that is something that we have yet to fully investigate. So this is what Gatenby originally wrote when he proposed this idea 5 years ago, which is eradicating metastatic cancers may be possible through a strategic integration of several therapies, each of which individually cannot achieve a curative outcome. And that is really a very nice summary of what we're trying to do here. And the basic idea then is you switch treatment even as the tumor appears to be responding well to the first therapy. That is what is unconventional. And there are already some preclinical and clinical trials in progress, and others that are in development that we hope to be running soon. So ultimately, what am I trying to do? I'm trying to bring these three components together. I've shown you stochastic spatial models, tree shape analysis, treatment models, and my big program is to try to combine all of these into better ways of clinical forecasting and smarter treatment strategies. And I think ultimately we want to combine all of these aspects together. So that's my last slide. I'll just thank all of the people who contributed to this work. There are a lot of very talented graduate students here, some of whom were based with me at City and some of whom were external. Where there are arrows, that shows how they have progressed from master's to PhD or PhD on to postdocs. And thank you also to my collaborator, Yannick Viosa, who has often been there to help me with particularly more advanced maths than I am capable of doing. And he's always a fantastic collaborator to have. And thank you to Vincent for ultimately developing all of this theory that we're now playing around with.
Thank you so much, Rob. It was fascinating. I really enjoyed learning about some of your work that has not been published yet. And please, if you have questions, post them in the chat. While we're waiting for questions in the chat, I do have some questions about the last part of your presentation. So first is kind of a technical question. I was a little puzzled by, you have a probability of tumor extinction due to, say, standing genetic variation. Typically, I would think that, you know, pre-existing resistance would lead to, you know, tumor growth or like relapse rather than tumor extinction. So how should I think about that?
This slide, you mean?
Yeah.
So, so R2 here is the number of cells resistant to the second treatment. R1 is resistant to the first treatment. Um, and the pi here is the probability of establishment. So this is the probability of them surviving.
Right.
So the larger these population sizes, the smaller the probability of extinction is going to be, right? Because it's e to the minus
Okay, yeah.
population size. So
Yeah, okay. No, I think I understand where you're getting at. It was just a little the other way around from what I was expecting. But that was just like a small technical question. I have a more general question about this approach of multiple strikes with different drugs. Um, I've done some work a long time ago on combination therapy, and somehow the takeaway there was that you really need combinations to get to cure. So how far do you think can this sequential approach uh, get us? Would it ever lead to cure, or would it just prolong survival, which is also great?
So yeah, that's a key question is would it be better just to apply these two treatments together initially?
Right.
We find that at least with these assumptions in this kind of model, no, it is better to apply them sequentially because applying them at the same time, you do not get the advantage of the stochastic extinction. So in the parameter ranges we're looking at, extinction is unlikely through unless you can get the population to be very small, because we're assuming there is pre-existing resistance. So your first treatment is very likely to, well, it's inevitably going to fail unless you can get that initial R1 population to go to zero. And the only real way you can do that is if it's extremely small. If it's not, then inevitably you will get bounce back. And so the main concept here is that by driving the population to this very small size, that's where it's susceptible to stochastic extinction. And that's when you want to hit it. That's why this works, which I realize now I didn't explain very well. The whole idea here is to exploit this susceptibility to stochastic extinction when the population size is very small, because then if you hit with the second treatment, then when the tumor is at its weakest, you have some chance that the population will go stochastically extinct. And that's not captured, of course, in ODE models and indeterministic models. And
Great. Yeah.
if you apply the two treatments together, you do not capitalize on that effect.
Yeah, that's great. That I'll think about some more, but that sounds fascinating. We have some comments in the chat. Matthew Osmond is explaining what that probability I was asking about was. Thank you. And then we have a question from James Garney. He says, "Hi Rob, good to see you. Are there any antagonisms, drug A resistance trades off for drug B in the models?"
There aren't. So we're assuming that these treatments are completely independent. But indeed, it will be very interesting to look at what happens if these treatments have antagonistic or synergistic effects. And that, yeah, and so there are many other model variants. So this is why we want to continue looking at these kind of models, and what we've tried to do here is the kind of paradigm case and really analyze that, and then we can see a lot of different ways it can be extended. But yeah, that is it would certainly help if you had some relationship between the drugs.
And we have another question from James. Does the population size matter at the stochastic extinction, or maybe the variation in the population?
What we find is it's really down to, so what you see here is the critical thing here is how large is the tumor when you switch. And you can see it's a very steep decrease here. So it really is critically down to that population size because of this stochastic extinction effect. In terms of variation, you would have to go into the supplementary material. So we there is a lot of nice analysis there of the properties of these populations, particularly that Srishti and Aman did. So I'm not sure that we actually plotted against the variance of the population size, but I think you would certainly find clues to that kind of analysis in the supplementary analyses.
Okay, more questions. So from Adam Palmer, how feasible would it be to scale up this model to regimens of 3, 4, 5 drugs as are common in some clinical regimens?
So analytically, it's fine. We can see how we would do that. It becomes, of course, more complicated because you now have to try you have to track a lot of different resistant populations. So here we've got three, right? R1, R2, and R12. Now, if you've got many different treatments, potentially you've got all those different combinations of resistance to all those different drugs. And you can make assumptions about those mutation rates, the cost of resistance. Potentially, you've got a lot of parameters to deal with. So that is one complication. In terms of simulations, we always run into a problem of simulating very large tumor sizes. But we do have some ways of getting around that. So I think it is feasible for us to do that. And it's certainly something we're proposing to do. But we haven't managed to do it yet because there are some issues that we have to grapple with first. But yeah, ultimately, I think that is the way this could work, is that this is not just two strikes. This is multiple strikes on the same idea.
Another question is from Rui Bojang. Do you know how these trees are inferred? I think he's referring to trees from the data. I think lineage tracing typically assumes a stochastic model like birth, death sampling, or coalescence.
So the original trees I showed you in the first slide I showed from TracerX, the method of tree reconstruction, I think it would be fair to say, was quite crude and quite simple. The method that is used in this latest paper by Frankel et al. Seems to me to be a very nice one. And there's an entire paper devoted to that method of inference for inferring these trees, which you would be able to find, I think, from the references in the Nature paper. So I believe that it's not my area of expertise, but it seems to me that they now have a very nice statistical model for inferring these trees that gives you a pretty robust idea of the tree shape. So there are some caveats. Yes, these are not necessarily ground truth. We can't tell exactly what the true tree is, but the other advantage now is that it's a very large cohort of 400 odd patients. So we hope that all those problems, if the trees aren't quite right, the method, the nice thing about our indices are that they do have this robustness. So if you get the trees a bit wrong, it shouldn't make a huge difference. And that's something we test by saying, what if we merge small nodes together, or we shrink short branches and things like that? And we find that our indices don't change much. I guess what we could also look at is if we run an inference model and we have multiple candidate trees, how much does our index vary between those different possibilities? We haven't done that yet, but that's something else we could do.
Great, thank you. A question from Alana Sholokhova. Great talk, thanks. In the tree balance segment, it looked like 0.85 is a balance cutoff for lung cancer survival. Why 0.85? How much does that vary between cancer types?
Yeah, so Kim, so we had so many discussions, Kim and I, about the fairest way to divide cohorts. And then we actually found a nice paper that describes this problem of setting cutoffs for cohorts. And there is no easy answer to how to do it. What we settled on was something that's a fairly round number, and that also splits the patients into reasonably equal groups, but also that is, so what we knew is that stage and grade is effective for stratifying these patients. So we knew that there was a small number of patients with very poor predicted survival and a larger number with better predicted survival and so on. So we chose cutoffs that meant that the group sizes we got, these 3 group sizes, were fairly similar to the ones you get if you use stage and grade, because that allows a fairly reasonable comparison with what stage and grade tells you. and it gives a kind of honest evaluation of our method. But what I want to stress is that this is just for illustrative purposes. The real test is what happens if you use your variable as a continuous variable and you don't split into groups at all. You say, how well does it predict survival as a continuous variable? And we found, what Kim found is that it is highly statistically significant if you do that, which is less easy to illustrate on a plot like this, but it does hold up. So the cutoffs don't matter too much. Kim tried various cutoffs and you get similar results. It's not that we really cherry pick the cutoffs.
And our final question is from Kit Gallagher. Thanks for the interesting talk. Are there any differences between drug 1 and 2 in multi-strike therapy, such as differences in efficacy, and could this affect the ordering of sequential treatment?
Yes, so I do have a supplementary slide on that as it happens. So this is looking at what happens if the first treatment and second treatment have different efficacies. So delta 1 is the efficacy of the first treatment. The higher it is, the more effective this treatment, and delta 2 of the second treatment. So here you put the more effective drug first, and in the second case, you put the more effective drug second. And you can see the differences here. So treating after the nadir, there's not a huge amount of difference in these two scenarios. But treating it before the nadir, you do see some difference. Now, if you compare these back to the slide I had before, if you just kind of visually memorize that before I flick back to the other slide, there's not that much difference. Here we have our default parameters, which are either both equal 2.0 or both equal 1.2. So this is high treatment efficacy for both. This is low treatment efficacy for both. So we looked at all these pairwise combinations, and this is what I mean that in this conclusion here, that the higher treatment efficacy is not necessarily all that beneficial. You can use less effective drugs, and by combining them and timing them well, you can still get very nice outcomes. So this is a good way of using drugs that perhaps usually in the clinic, they might say it's not going to be, we wouldn't normally treat with this because by itself, it would never cure the tumor. And what we're saying is essentially what Gatenby said here, which is that the strategic integration of these therapies, each of which individually cannot achieve a curative outcome, might still eradicate the cancer.
Excellent. Thank you so much, Rob, for being our first speaker. Thank you to everyone watching for the great questions. And I'll see you in the next installment of the MCMC seminar.